Hello, everyone, and welcome to the Lytham Partners Spring 2023 Investor Conference. My name is Ben Shamsian, Vice President of Lytham Partners. Thank you for joining us today for our oncology panel titled Advancements in the Fight Against Cancer. We are joined today by two executives from two great companies from the industry to get some insights on how they see the space. I've gathered some topics of interest to investors and hope to learn greatly from our panelists. Joining us today are Gerard Michel, CEO of Delcath Technologies, ticker DCTH on the NASDAQ, as well as Dr. Anatoly Dritschilo, CEO of Shuttle Pharmaceuticals, ticker SHPH. I want to remind everyone that our management teams are available for one-on-one meetings. If you have not already signed up, please send me an email at shamsian@lythampartners.com. That's SHAMSIAN@lythampartners.com, or visit lythampartners.com/virtual and click the one-on-one meeting request button. Before we begin with the discussion, I would like to have each of you introduce yourselves and provide a 5-minute background on your companies. Gerard, we can start with you.
Sure, and Ben, thanks for having me, and Anatoly, good to virtually meet you today. So Delcath Systems is a company that has a combination drug device that treats liver mets. Now, it's important to know that for many, if not most solid tumors, it is the metastatic disease, not the primary tumor, that eventually is the cause of death for patients. The liver is one of the most common sites of metastatic disease for many patients across multiple types of solid tumors. And besides, brain mets are probably the most difficult to treat. Resection is only an option for maybe about 15% of patients across all the different tumor types. Systemic chemo is not very effective at tolerable doses, and even immuno-oncology agents actually lose their systemic efficacy when liver mets appear because the liver induces systemic immune tolerance to any antigens that are presented to the liver. There are about 200,000 primary and metastatic liver tumors per year. This combines colorectal, pancreatic, breast, intrahepatic cholangiocarcinoma, and a whole host of other tumors, as well as primary liver cancer. It's a very large unmet need. Various organs in the body have very different dose-limiting toxicities to chemotherapy, with hematological toxicities being the most common dose-limiting toxicity for most chemo regimes. So your blood marrow often gets wiped out, called myeloablative. The working cells of the liver, hepatocytes, are quite robust and can handle far more, 5 to 10 times, depending on the chemo agent, the maximum tolerated dose of commonly used therapeutic agents systemically. The drug we deliver, melphalan, is delivered directly to the liver, but we limit the systemic exposure by our hepatic delivery system, or HDS, which is the device component of our combination product. The HDS is comprised of a double balloon catheter, which blocks the hepatic venous blood from systemic circulation and transports the blood into an extracorporeal circuit, or outside the body, where there is a specialized filter which absorbs about 85% of the melphalan. So the blood is returned to the patient via another catheter placed in the internal jugular. In this way, we can deliver chemotherapy to the tumors at much higher levels than you would otherwise be possible to do so systemically. Our target is eventually to take this to numerous different cancer types. Our first tumor type is an ultra-orphan metastatic ductal melanoma, a tumor that starts in the eye, and about 50% of the cases eventually become metastatic, mostly to the liver, and that's generally what patients succumb to.
All right, thank you. Anatoly?
So I'm basically an academic who has advanced to develop some of the discoveries and research that I've been doing over the past 40 years, actually working for Georgetown University as chairman of radiation medicine and Lombardi Comprehensive Cancer Center, and for the Georgetown University Hospital, where I've been chief of radiation oncology, treating many patients with radiation therapy. The issue that is at hand is that we have a great way of delivering radiation. It's a very effective type of treatment, but there are limitations to what can be done. Although the dose that we can deliver to any location inside the body is dictated by the technology, the complications and the damage to normal tissues is limiting in many areas. So we can't get the dose that we need to get into. So the solution for that is to use radiation sensitizers and to use immune oncology, as a combination. So our solution for brain tumors, which we've targeted as one of the most pressing problems in our field, is to add a drug that is taken up by the cancer cells and then makes the cancer cells more sensitive to the radiation effects. So we can prolong survival in patients by killing more cancer cells, imparting more damage to the cancer cells. The second area would be at the back end of after treatment. What happens is you have damage to the cells, which means that antigens come to the surface in the tumors, and you get your innate immune system is activated. And what we have developed is several drugs called HDAC inhibitors, histone deacetylase 6 in particular. And this is an enzyme that if it regulates the immune response to cancers, and that we're able to stimulate that and prolong the response and treatment of the cancer cells after radiation. So those are the two ends that we are targeting. The front end, which is the sensitizer to make the radiation more effective, and then what is left in any residual, which is limited by total dose that you can deliver on the back end to stimulate the immune system.
All right, thank you. I know you both alluded to it slightly on in your sort of opening remarks, but I want to get to the problem that each of your technologies is looking to solve. Dr. Dritschilo, I'll begin with you. Specifically, what is the issue here that you're trying to solve?
Well, the problem is always to get more damage into the cancer cells than you get into normal cells. I mean, it's true for our product. It's true for, Delcath's product. You're always trying to target the tumor preferentially to the normal tissues. In situations where the tumors are located and can be isolated, I mean, surgery is very effective. Radiation therapy is very effective. It's when the tumor is located inside the brain, or let's say inside an area where you have lung, heart, where you have intestines surrounding. These tissues are more sensitive to the treatments, and they are dose limiting. They prevent you from going to the curative doses. So by making the effectiveness greater by sensitizing, that would be the goal of the treatment to improve the response of the tumor.
Got it. Gerard?
Sure. So I think when we're talking about liver metastases, there are two treatments out there. Well, there's resection. Some people do radioembolization. Some people use coils where they can put magnetic fields in that heats up the tumor and kills it. There's a whole host of ways of treating tumors in the liver, as long as you can access it generally with a catheter. But those are all great for focal disease. So if you have a tumor you can see and you know it's the only tumor, you can use something called transarterial chemoembolization, PACE, which are beads that occlude the arteries that are leading to the tumor, and they also elute chemotherapy. And those generally have pretty good response rates for those focal tumors. There's another treatment called yttrium-90. Two companies produce those. They're radioactive beads. Generally, you do a lobe by lobe treatment with that in the liver. And that's fairly good at treating local disease as well. But if you have diffuse disease throughout the whole liver, whether it's a miliary pattern, which is very, very diffuse, or even if you don't see a miliary pattern, for many diseases, when you have liver mets, if you take a biopsy of the liver in what looks like apparently healthy tissue, you'll look at it under a microscope, that'll look apparently healthy as well. But if you use hybridization probes or a number of other technologies, you'll find that there are many, many micromets for many different tumor types. This is common. And what's happening is those somewhat dormant mets will eventually pop up and become active again. So whether it's miliary, where you have lots of little pea-sized tumors that are hard to image, or whether or not it's even not that, but you have focal disease, generally you have mets either pending or in, or actually there, they're just not available radiographically throughout the liver. So you need a way to treat the entire liver, not just the focal disease. Otherwise, you're playing whack-a-mole. And eventually you have to stop playing whack-a-mole with the existing therapies, whether it's Y90, because you're really limited to 2 doses of that because it's very hepatotoxic, or whether or not it's TACE. By the definition, you destroy the vasculature whenever you're treating the solid tumors with TACE. So our treatment treats the entire liver. You don't need to see where the tumors are. So we solve that innate problem. We're also able to, because the hepatocytes are quite robust against melphalan, so we do very little hepatic damage. We're able to treat multiple times. Our phase 3 trial limited to 6 times. We've had patients in Europe treated up to 8 or 9 times with the disease. So we can treat the whole liver. You don't need to image where the tumors specifically are, and you can do it repeatedly. And it has very, very low hepatotoxicity.
That's great. Gerard, you alluded to some of the alternatives that are out there. Can you speak a little bit more to those, and what is it that you bring to the table that's technologically superior to what's already?
Yeah, again, with TACE, you have to see the tumor to treat it. And usually if you have 1 or 2 radiographically obvious tumors, you're going to have multiple tumors. Not always, but that's generally the case. So with TACE, you have to be able to see the target to shoot it. Ours treats the entire liver. Y90, similar issue. It treats a bit more broadly. You can treat a whole lobe with Y90, but Y90 is highly toxic, and you're really limited to two treatments. So we can keep going in there, keep disease at bay, and we don't need to see where it is. Longer term, we hope to take this treatment to more of almost an adjuvant setting for very high-risk patients, for example, colorectal patients who may still be ctDNA positive after resection, and ctDNA positives in the liver. We'd like to see patients being treated there, and hopefully we can prevent radiographically evident mets from showing up.
That's great. Anatoly, talk about the alternatives that are there for you, and sort of what makes your product and your technology superior.
Right. So the use of sensitizers is not a new concept. There are drugs that are out there that have single agent effectiveness. So one of the primary ones is 5-fluorouracil. It's a drug that is used all the time in treating GI cancers. It tends not to be a curative drug, but it certainly does put patients into remission, and it prolongs their lives. So this is something that is used. That drug has a side effect of sensitization. So therefore, many protocols for treating cancers include the addition of radiation with 5-fluorouracil. There are several other drugs, including things like cisplatin and other, let's say, agents that have single agent activity that were approved for that activity, but then as a side effect, they're used off-label by physicians to sensitize. So sensitization is something that's recognized by oncologists and is used as much as possible. The problem is that these drugs have side effects. There's only one drug that was actually approved directly as a radiation sensitizer, and that's a drug, Erbitux, which has been something that is used in head and neck cancer. But again, that has a targeted direct effect on other, let's say, biological markers. So it has a lot of reaction that occurs when it's used in head and neck cancer. So the drug that we have that we're trying to advance in our phase 2 clinical trial is ropinirole, which is a sensitizer only, meaning that if you give the drug to the patient and don't irradiate, you get no effect. So it's a true radiation sensitizer. When you combine it with the radiation, the drug tends to get into the DNA and basically allows for greater damage to the DNA, more double-strand breaks, and with that, more cell kill. So that's a true radiation sensitizer. There are several drugs like that, that are in the laboratory level, but basically this is one of the benefits of a true sensitizer. The other drugs that we have are the HDAC inhibitors, which regulate cell processes by regulating DNA gene expression. Those have to be used in combination with other treatments also in order to get a true benefit. There are several that have been approved for use in lymphoma, and they do have some direct effects. But the HDAC6 targeting, which is a very selective enzyme that we target for immune modulation, those are very, very selective, and they don't affect the other categories of HDAC molecules.
All right, thank you. A question we get from a lot of investors is sort of market size, TAM, sort of what are we addressing here in terms of, sort of the bigger picture, right? So can each of you talk about the addressable market opportunity that your products, your solutions provide? Dr. Dritschilo, I'll begin with you.
Okay, so there are two basic markets. One is the initial market that we're going after is the most aggressive type of brain tumor that there is, and this is the methylation negative IDH wild type brain tumor glioblastoma. There's about 10,000 patients with the glioblastoma brain tumors that are diagnosed every year. About half of them would fall into this most aggressive type. So that's a market size with a very great need, and that's what our clinical drug development is working toward. In terms of the HDAC inhibitors as sensitizers and as stimulators of immune response, we have several patented agents that we're developing in preclinical work. The market is much larger. And also the market is larger for the ropidoxuridine if we get initial validation and approval for the brain tumors. We would then be able to pursue through clinical trials other indications where there's rapidly growing tumors that take up the drug and they're still localized. They cannot be systemic everywhere. They have to be localized in a regional area for us to be able to treat them. So that's about 1.8 million patients with cancer diagnosed every year, according to the American Cancer Society. About half of them are being treated for what we call curative intent. The other half are treated for palliative intent, where the disease is too advanced to really cure the patient. But you can provide benefit by putting them into remission and prolonging their survival and get value that way. And then out of that, 50% that are, let's say, local regional, again, you go about half again, and that comes down to about 450,000 patients with disease that can be treated with radiation in combination with sensitizers. So the market is very large. The agents that you would use may be targeting specific targets and specific tumor types, so there is some personalized medicine associated with it, but some of these agents certainly overlap. There are multiple cancers that can be treated. So it's a very large market, and it's a matter of demonstrating through the clinical trials that FDA approval for commercialization is the barrier that has to be overcome, and appropriately so.
Thank you. Gerard, you've been specific about the TAM in previous presentations. Help us understand how you see the market.
Sure. So I think, as everyone knows, you can have a great product for theoretically a broad range of cancers, but regulatory approval is, on an indication by indication basis. I had mentioned that there are probably about 200,000 patients a year It's an incidence number who get diagnosed with liver-dominant disease. In other words, the liver mets are likely what's going to kill the patient. But we have to start, in a specific indication. The indication we've been working on for quite a few years is metastatic ocular melanoma. That's a tumor, as I mentioned, that starts in the eye, and that about half of the patients eventually it metastasizes. Most of the time, 90% plus of the time, it's a liver-dominant disease. So it's probably about 2,500 patients in the U.S. and Europe, 2,000 to 2,500 in the U.S. and Europe. If we focus for a moment on the U.S., because that's really where we're going to make the bulk of our revenue and profit, and there are probably about 800 patients who fit within our TAM. Now, these patients don't have a lot of options. There's only one product approved for them. It's a product called Kimtrac. That is a modest extension of life. They've shown several months. We think our data is comparable, but comparable in a more advanced patient population, and that our trial was done in a mix of first, second, and third line patients. They were all done in first line patients. Their TAM is probably about 250 million based on how they've priced. If we priced on a comparable basis, our TAM would be close to 500 million. So that's a reasonable TAM. Longer term, though, we want to be able to treat patients across multiple different cancer types. Then we're talking in the billions. It's easy for CEOs to throw around numbers of B's. But in the short term, next couple of years, we're talking about revenue potential in the hundreds of millions of dollars. And Kimtrac, again, approved in the U.S. for only about half of this patient population because they need a specific allele for their product to work. They're already at a run rate of well over 100 million in about after about 3 quarters to 4 quarters of being on the market. So we're hopeful with twice the TAM, we can do a similar uptake.
Got it. Thank you. Let's talk about the FDA. You're both in the process of FDA approval, different stages. Tell us about your experience there and what you've seen the FDA become easier or more difficult to navigate. Gerard, I'll begin with you.
Sure. So it's tough. You read through the industry literature and people take trends based on a couple of immediate events or recent events and draw conclusions as to whether it's easier or harder. It's hard for me to say. I could say over the long term, it's certainly costing more and taking more time. But I say that based on being in the industry for about, I hate to say it, about 35 years at this point. What is considered industry standard and what is expected both preclinically and clinically generally only moves on one direction on a net basis, and that's towards increased complexity, rigor, and therefore it leads to greater cost and time. The FDA is always trying to do the right thing, whether it's to push, and I've mentioned some more recent initiatives. They put some guidance out towards it's either more diversity in the clinical trials, a recent guidance on finding the optimum dose instead of a dose which is safe and effective, inevitably adds cost and time to development. But for our program right now at this stage, I don't think there are any emerging trends that will directly impact us.
Thank you. Anatoly, on the FDA process?
So we're at an earlier stage. We're at stage 2, in terms of clinical trials, and it's a simple solution. You do what the FDA tells you, and you don't have a lot of debating ability. However, certainly you can get input from the FDA and some guidance to make it efficient. After all, they do want to see these drugs developed, and I don't think it's an adversarial. But for the FDA, safety of patients, comes first, and then the efficacy and development are things that they want done, but in the face of safety for the patient.
Got it. Thank you. Let's talk about the cost of the system. Can we talk about the value that your solutions bring to addressing the overall cost in healthcare? Dr. Anatoly, I'll begin with you.
Yeah, so there's value in the following ways. There's direct value in terms of if you prolong survival and you improve, let's say, the cure rate. This is something that is pretty easy for people to see. But there's also value in preventing the progression of disease and complications that require additional treatment and surgeries and management. So you have a couple of things that have to be balanced. In general, the drugs that we offer, increase the sensitization, and we don't. We look to decrease the number of, let's say, complication-free survivals and cure rates with patients by using the sensitizer. So this is something that is pretty easy. It's a small molecule drug. Also, it's not real expensive to make, so that it's not going to be a very expensive, drug to give to patients, such as a biological, perhaps, where you're dealing with something that's hundreds of thousands of dollars. This we're talking about in the range of, I think, in our S1 we did the calculations based on about $9,000 per course of treatment, which would be typically for a brain tumor would be a course of 6 weeks, 30 treatments of radiation, and you would give the drug concomitantly with the radiation treatments. Those are the numbers. So there is value. If we can prolong survival, can improve the local control without causing additional damage. I think there is some benefit, because you can with sensitizer you can use lower doses for let's say critical parts of the brain. For example, you can't treat the central part of the brain called the thalamus to very high doses. You're limited. You can treat other parts of the brain to, let's say, the full dose, but these areas around the optic nerve, your ability to see and so forth, have to be protected. So usually it's a lower dose, and if you use a sensitizer with it, that lower dose may offer a benefit of preventing disease from progressing in those areas.
Thank you. Gerard, you've been a little bit more specific on sort of the cost, the value that you see out there. Help us walk through the math a little bit.
Yeah, and it's sometimes you have to be specific whether you want to or not, because we're playing with other people's money, and we have to convince the investors they're going to get a return. I think in terms of value to the system, there are lots of ways to calculate that. And I've been through this many, many times, whether it's, is the average of cost per life going to be a penny, 10 cents, 20 cents, whatever, all the way to, what is the cost for quality adjusted life year. I think to answer your question, Ben, I think for us, there's a marker out there, a product called KIMTRAC, that you need to spend about $500,000. It's an ultra orphan product, maybe 300, 400, 500 patients out there for them. And they intend to take this platform, their bispecific antibody, into other tumor types. And you kind of think of this as it's costing one disease state as an investment to move it forward into many others. But they have to charge about $500,000 before they know whether or not the treatment is working. The reason for that is it's an immuno-oncology agent. And with immuno-oncology agents, you have something called pseudoprogression, where you'll often get actual expansion of the tumor radiographically when you look at it due to an immune reaction. So you really can't tell if the tumor is progressing or you're actually just getting a positive response. So you treat through progression. You need 2 confirmed progression reads before you go off that therapy. So that's quite a long time to be on a therapy when you really don't know if it's working. And probably for the majority of patients, they aren't getting a benefit. Smaller percentage of patients are getting a profound benefit from what I read from that product. You got to use it for quite a while before you know. Ours is a more classical cytotoxic. You'll know pretty much after the first dose or the first scan, after some doses, whether or not you're getting a result. So we'll know a lot faster. And even if we price at a comparable basis as KIMTRAC, it'll probably be about half the cost or less before you know whether or not you're getting a benefit for the patient, both from the cost perspective, as well as, you don't want to give the patient any medicine that's giving them any benefit. I would note that, hundreds of millions of dollars have gone into this company over time. Our hope is to price it in a way that does bring real benefit both to investors and to patients over time. And hopefully we'll use this in multiple different types of cancers beyond just this ultra orphan category, which does bring different economic constraints in terms of how you have to price it.
Got it. Thank you for that. Let's move on to some of the challenges that you have to endure. The small company, pre-revenue stage for both of you. What are some of the challenges that you either have gone through or you're currently going through, and sort of how are you managing those issues? Gerard, I'll begin with you.
Yeah, well, our challenges are quite public in that I've been very open and transparent about them since I joined a little over about 2.5 years ago. We've been a company with a complete response letter roughly a decade ago. This was a company where there was a great idea from a number of surgical oncologists. It went through a number of management teams that, frankly, in hindsight, I can say this: Monday morning reporting back, just kind of dropped the ball and made some big errors. I came in after the company was recapped. It was a skeleton crew. It's still a lean crew. And tried to turn the ship around and take a product that does work. And now, since I've come, a set of data that clearly is compelling, and get the product approved. So I think a lot of legacy mindset outside of the investor community was probably one of our biggest challenges to deal with, and also to track capital. That has not been easy. I'm thrilled that we have a number of top-tier investors involved now in our last round. Vivo, BVF, Logos, Stone Pine, all our top-tier biotech sophisticated investors. It was great to get them on board. So I think we're beating that challenge of kind of a legacy mindset out in the investor community. And for us now, it's with a small team answering the FDA information requests, which are coming in now as they're reviewing our filing and approaching the PDUFA date on August 14th, and prepping for launch.
Got it. Dr. Dritschilo, some challenges ahead of you.
So we're a very early company in terms of being public, and there's clearly a learning curve about how you become public and what people are looking for. Just because you have a good idea and you have some data that's in the laboratory doesn't necessarily mean everyone's going to recognize it. They run the business plans, they make decisions that basically may or may not be relevant from your point of view, but they certainly are relevant from investment point of view. So we're pretty early, and I recognize that the key is to meet the scientific milestones, which I've done all my life, through my academic and clinical practice. I've treated thousands of patients, and I've been director of a cancer center. I've been director of a hospital and a department. So these are things that you have to face on a regular basis and just deal with them as they come along. So these are the challenges. Now, in terms of being able to reach your milestones, you have to have enough of a runway in terms of capital to do that. And I have focused on that. I've worked with a very effective chief financial officer, somebody who I worked with for about a number of years back at when we were at Lombardi Cancer Center. So I know that he is capable and manages that part. So I have very good people in those areas. When we're dealing with drug manufacture, I have people that I trust, and I've developed a relationship over a number of years. So these are all things that have to be addressed. They're there, and I think it's part of doing business.
All right. Let's talk about partnerships. As a relatively small healthcare company, partnerships are sometimes an avenue many companies take. Love to hear your thoughts, your philosophy regarding partnerships, some of their benefits and drawbacks. Gerard, I'll begin with you.
Yeah, obviously, partnering is a key part of the biotech industry. and it's a key source and strategy for non-dilutive capital. But I ran corp dev for a number of smaller biotech companies over the years and did that in big pharma for a while. In our particular case, we're neither fish nor fowl. Drug companies look at us and they kind of think this is a device company. The device companies that are interested in us are more interventional oncology. There's definitely a set of those that should be interested. But I don't think they generally don't do licensing deals. They'll do maybe distributorships. And usually the big deals on the device side happen after you're approved and you have some revenue and proving yourself. Because it's usually more bolt-on acquisitions where they're really focused on accretion and dilution. And they want things to be accretive as quickly as possible. And they want to be able to do the math. In the biotech arena, it's much more commonly that people will partner very early on, often without even proof of principle, either a platform or after there is some proof of principle. And it makes a ton of sense to try to do, a European deal or something, or carve out one or two indications or molecules if you have a platform that'll keep spinning things out and that'll help pay the bills for a while. But in my particular instance, I think it's more of an issue where we'll get launched, approved, and I'm not predicting anything, nor am I stating this is the strategy for the company per se, but it's more likely we would just be bought outright than do a partnership deal. And the best way to get sold is to pretend that no one's ever going to buy you and just execute. And, that's our strategy right now.
Got it. Anatoly?
So we work at two levels here. So one level is the academic relationships that we have. And, I have a number of colleagues in several of these, academic institutions where we can do the clinical trials, in order to complete. So for the ropiduxuridine for the treatment of brain tumors, that's what I want. I want the best centers that are available, academically oriented, to do the clinical trials. So I know that it's done in such a way that it's not diluted out by, let's say, poor accrual or poor, let's say, patients that don't exactly fit and basically, can destroy a small clinical trial. And we're looking at a small clinical trial, and we'd like to, magnify our relationships with personal relationships we have, and to expand those. In terms of the ability to partner with the outside, I agree. You need to have something you can license out, and some platform technology that perhaps is of interest. So when we first started our company, we spent the first 5 years basically doing drug discovery. That's all we did was say there are 12 enzymes in the HDAC system. And we have half a dozen patents that we have developed. So we do have class 1 and class 2 HDAC inhibitors. These are preclinical things that we're developing. We do have the HDAC6 selective inhibitors. So we went in a direction that the pan-HDAC inhibitors, the ones that affected all 12 enzymes, were just too toxic to use clinically very effectively. And by focusing on the selective properties, we were able to develop a number of agents that we will be working on. As we develop these, we will be looking for partnerships. We will be looking for other companies that may have an interest in sharing this, especially if we are able to advance some of the immune stimulatory factors that we can. We do have a strong patent portfolio that is wholly owned by the company. So these are patents that we don't license them from someone else. We own the patents. So this is something that I think is different about our company than other companies.
All right, thank you. And final question. I'd love each of you to tell me something either your potential customer base or your investor base currently may not fully appreciate about your company, either your company or the technology. Dr. Driscoll, I will begin with you.
Hey, so the concept of radiation therapy and to improve radiation therapy is a highly nuanced area, meaning that even physicians don't really truly understand everything about it, and they don't understand what happens, and you basically have to educate many people who are in practice and taking care of patients. It's not something that people focus on, but they do know it exists, okay? But it's not something that is out there in strength. So we have to educate people about where our niche is and where we can improve things. Yeah, so that's one thing. So, but in terms of let's say investors, we're a biotech that it's going to take time for us to finish a phase 2 clinical trial to reach the next major milestone, which will be plus or minus on the clinical trial. We have a drug that we're going to be doing the IND enabling and preclinical testing to get to phase 1. Again, that takes time. You have to manufacture the drug, you have to formulate it, you have to test it in animals for safety and efficacy, and then you have to basically do a phase 1 clinical trial. Again, you're talking about a package that's about 1 year to 2 years that has to be dedicated just to get to that point. Investors think in much shorter time periods. So to some degree, the best investors are the longer term, let's say, institutional investors that may have a longer time frame. Unfortunately, you take basically what you can get in the environment. This is a tough environment right now, for raising capital. We've been fortunate to raise enough so we do have a runway, and I think that most investors that would come in at this point in time would have to hang on to the stock for, let's say, longer than 1 year to feel comfortable about getting somewhere with it.
Thank you. And Gerard?
Yeah, I think you probably choose a number of things that people don't get. I mean, I think the first thing is, how many people are paying attention to you. There is just a technical, and when I say technical, I mean a capital markets, a technical aspect of small microcap companies where the more sophisticated investors aren't going to look at you because they have large amounts of money to put to work, and they have constraints about, it's not worth their time, or they're prevented from buying into smaller companies. So then you're into family offices and such, and companies that are our size. There are thousands of them out there, and it's hard to get share of mind with these smaller funds. And it's not who the bank service, whether in it's non-deal roadshows with analysts or other techniques. It's tough to rise above the crowd. So that's one constraint. And I think the only way to get beyond that, besides working with quality folks like yourself, Ben, is to just deliver. Don't go bankrupt. Keep the money in the coffers and deliver, and eventually people will pay attention as you put clinical or scientific points on the board. More specifically for us, I think what people don't get is that we have what appears to be a very pedestrian set of technologies that we're using together. It's typical cytotoxic chemotherapy, a double balloon catheter, and a filter. What's so magical about that? But it works. There are a lot of simple things that work, and this should work in multiple tumor types beyond this small one we have. But the other aspect of it is, if you couple it with newer technologies or technologies that are out there that aren't currently being used, it becomes a much more powerful potential therapy for patients. And I'll name a couple that anybody's following us has probably heard me talk about before. One is immuno-oncology agents. As I mentioned early on, the liver induces systemic immune tolerance to whatever antigens are chronically expressed in the liver. And that's because the portal vein, which wraps around the gut, drops into the liver. If the liver didn't do that, we'd all be walking away with IBD or some type of inflammatory bowel disease. It's one of the reasons hepatitis is so difficult to get rid of. Now, what that means is that both when you have liver mets, there's some evidence that tumors can get more aggressive systemically, but there's definitely strong evidence that immuno-oncology agents stop working. Now, people have tried in animal models using low-dose radiation delivered to the whole liver, basically to wipe out the various immune cells in the liver, reset the microenvironment. And what they've seen is immuno-oncology is regaining their efficacy. Now, we're seeing a signal like that in the Chopin trial at Leiden University in the Netherlands, where we are alternating immuno-oncology agents, which typically don't have great response rates for ocular melanoma, with our product, where we're essentially wiping out all the immune cells, Tregs, macrophages, et cetera, and we're seeing profound response rates and very, very durable response rates. Now, we'd love to see that tried in hepatocellular carcinoma, where IO agents in this country are second line per NCCN guidelines. I'd like to see it done with microsatellite unstable or mismatch repair CRC, where IO agents are our first line. I'd love to see that. And that's a cancer type where you frequently get a lot of liver mets. So I think in combination with IO agents where there are liver mets, there may be a very, very dramatic potential. Secondly, patients, as I mentioned before, often have micromets that are not detectable. And if you could detect it, we'd be with a ctDNA from the hepatic vein, or whether or not you could do biopsies using hybridization probes to see mets that aren't visual even on histological sample as you use the probes. Could we use, in cases where the patient's positive, could we use our product in an adjuvant setting? Close to 60% of all CRC patients end up with liver mets. Most of those you might be able to, hopefully you could detect those far earlier with some of those technologies that I just mentioned. And if you used our product in an adjuvant setting, I think everybody knows the chemo works very, very well when tumors are very small. Perhaps we could make a dramatic difference in those patients' lives. So I don't think people recognize how we could use this tool in combination with those and other technologies that are out there emerging to really make a difference in patients' lives.
All right. Well, thank you. Fortunately, that's all the time we have. I want to thank our panelists as well as everyone listening. If any investors would like to get in touch with any of our panel companies, please reach out to myself or any members of the Lytham Partners team. Thank you and have a good day, everyone.
Thank you very much.
Thank you, Ben.